Here are 5 USMLE-style questions focused on tRNA structure, charging, and the accuracy of amino-acyl tRNA synthetase. Question 1. Which of the following best describes the function of the D arm of tRNA? A. It is responsible for binding the tRNA to the ribosome during translation. B. It contains the TEAP-seq sequence, necessary for ribosome binding. C. It contains dihydrouridine residues, enabling recognition by amino-acyl tRNA synthetase. D. It serves as the site where the amino acid is covalently attached to the tRNA. E. It is involved in the hydrolysis of the bond between tRNA and the amino acid. Answer. It contains dihydrouridine residues, enabling recognition by amino-acyl tRNA synthetase. Explanation. The D arm of tRNA contains dihydrouridine residues that are important for the recognition of tRNA by the correct amino-acyl tRNA synthetase, which ensures accurate amino acid attachment. The T arm, choice B, is involved in ribosome binding. Question 2. A mutation occurs in the CCA sequence of a tRNA molecule. Which of the following is the most likely consequence of this mutation? A. Decreased aminoacylation of the tRNA. B. Inability of the tRNA to recognize the codon on mRNA. C. The tRNA will not be able to bind to the ribosome during translation. D. The tRNA will be unable to undergo splicing. E. The tRNA will be unable to attach the correct amino acid. Answer. A. Decreased aminoacylation of the T-planation. The CCA sequence at the 3' end of the tRNA is where the amino acid is covalently bound. A mutation in this region would impair aminoacylation and prevent the tRNA from attaching the appropriate amino acid, disrupting protein synthesis. Question 3. A patient is found to have a defect in amino-acyl tRNA synthetase, which results in an inability to properly charge tRNA with its corresponding amino acid. Which of the following is the most likely consequence of this defect? A. Increased accuracy of amino acid incorporation during translation. B. Decreased binding of tRNA to the ribosome. C. Production of mischarged tRNA, leading to the incorporation of incorrect amino acids. D. Inability to form peptide bonds between amino acids. E. Failure to bind mRNA to the ribosome. C. Production of mischarged tRNA, leading to the incorporation of incorrect amino acids. Explanation. Amino-acyl tRNA synthetase is responsible for attaching the correct amino acid to the tRNA. If this enzyme is defective, mischarged tRNA will be produced, which can result in the incorporation of incorrect amino acids into the growing polypeptide chain, leading to faulty proteins. Question 4. Which of the following sequences is located at the attachment site of a tRNA molecule? A. TC, 3,3 ACC5, C. Anticodon loop, D. 5'IAG3E 5'IUTR? B. 3D explanation. The 3-ACC5 sequence is the attachment site where the amino acid is covalently bound to the tRNA. This is the amino acid acceptor site, and it is essential for proper charging of the tRNA. Question 5. A drug is designed to inhibit the function of amino-acyl tRNA synthetase. Which of the following is the most likely effect of this drug? A. Increased protein synthesis due to more accurate amino acid matching. B. Decreased protein synthesis due to the inability to correctly charge tRNAs. C. Increased production of poly-A tail in mRNA. D. Decreased translation due to disruption of codon recognition. E. Increased transcription of protein-coding genes. Answer. B. Decreased protein synthesis due to the inability to correctly charge tRNAs. Explanation. Amino-acyl tRNA synthetase is responsible for charging tRNA molecules with their corresponding amino acids. If this enzyme is inhibited, tRNA charging is disrupted, leading to a decrease in protein synthesis because the ribosome will not be able to receive the correctly charged tRNAs for translation. These questions cover the